from a fertilized egg, how is it that a complex organism with brain and liver and so on can develop? And what can go wrong?
With these questions, Rudolf Jaenisch is leading a revolution in how biology is done, fusing embryology, molecular biology, and virology. He has dissected the process of epigenetics.
Epigenetic is the way the genome talks with the environment.
Born in 1942, Jaenisch began his career in West Germany, where he was raised. After he received his medical degree at the University of Munich in 1967, he became a virologist at the well-equipped Max Planck Institute. In 1970, he moved to Princeton as a postdoc.
I was really shocked. The laboratory was in an old building. The mold was coming out of the walls in the tissue culture room. But what really matters is not shiny instruments, but the atmosphere. There was just incredible excitement scientifically.
Jaenisch expanded his virus research to mice. He injected adult mice with a tumor virus that caused just one kind of cancer.
Why does this virus only make skin tumors but not brain tumors or liver tumors?
The technology did not yet exist to find out. Then he read a paper that changed his life. Philadelphia-based embryologist Beatrice Mintz had taken black mouse and white mouse embryo cells and mixed them together in a petri dish. The mixture of cells formed one embryo, which grew up into a striped mouse.
If you could insert the viral DNA into these early embryos, they would end up in the liver, in the brain, and in the skin. And then I could answer my question. So I was very excited. I couldn't sleep, so I called her up the next day.
She was skeptical. His boss, Arnold Levine, was more blunt.
Arnie said, "You are nuts."
Jaenisch spent months injecting embryos with viral DNA until he got mice that grew from the virus-treated embryonic cells.
I had expected I get mice with tumors coming out of the brain and everywhere. They didn't. They were totally normal.
There was no way at the time to know for sure whether the mice had virus DNA in all their organs. Jaenisch moved to his first faculty job at the Salk Institute in 1972. There, borrowing a colleague's new technique for testing DNA, he got his answer.
And to my surprise, yes, there was lots of DNA in these organs.
Though tumor virus genes were tucked into the mouse's genome, something was turning them off. It was an example of epigenetics, the key to cellular development processes and many cancers. Such epigenetic on-off switches can even get passed on from one generation to the next. In 1984, he joined MIT's Whitehead Institute in Cambridge, Massachusetts, where he has been ever since. Thanks to the work of Jaenisch and his colleagues, scientists can now design mice to model and treat many diseases. In 1997, Dolly, the first cloned animal, was grown from an embryonic stem cell. Jaenisch became an outspoken critic of reproductive cloning. He argued for therapeutic cloning. Rather than growing a whole person, cloning only the tissues needed to cure disease.
Therapeutic cloning was a great idea. So exciting.
But human cloning required human eggs to program genetic material into an embryo, an idea which was not practical.
The key issue was to learn how does the egg do it, and then do it without the egg.
Jaenisch's lab was one of the first in the world to figure out how to make adult stem cells, overcoming the practical problems associated with using eggs and embryos.
We showed that you can use therapeutic cloning to treat a mouse which had immune deficiency.
Jaenisch's work is paving the way for patients to be cured of diseases with improved versions of their own cells.
It's a game changer in the sense that now it is not anymore a pie in the sky type of thing. The principle is established. I think this will change not only how we study human diseases, but how we can treat diseases.
Rudolf Jaenisch was awarded the 2013 Benjamin Franklin Medal in Life Science.